on artificial intelligence and share you a little bit about a tool that my company has developed, which was started in the year 2017, so quite recently. So we're a startup company based in San Francisco in the US. And it's basically about the connection between plants, nutrition, and health. So I'm an employee of the company, Brightseed. So a lot of the bioactives and plant compounds are hidden, right? We don't know about them. We call them the dark matter, and there's clearly a lot to be investigated there. The published literature is less than 1% is described. So what's the 99% out there, or more than 99%? It's also interesting to note that a lot of the approved registered drugs find their origin in plant bioactives, whether it is the bioactive itself being used as the active in a pharmaceutical drug, or it is an active or a structure based found in nature, right? That is used. Aspirin comes from the willow bark tree, metformin from French lilac, and so there are so many other examples. The Forager tool, is an artificial intelligence tool that's won quite a lot of awards and identifies potential bioactives for known targets at a very high rate. I'll share you a little bit more about how the tool works in one of the next slide. The tool has developed visibility in more than 2 million plant natural compounds, and that's a lot more than what is known in the published literature at the moment. And we're actually on target in 3 or 4 years to have developed a plant database of plant compounds that encompasses all the plants around the world, almost all. So that is quite significant. We have a number of partnerships. We work together in academic partnerships. We work also with the Bill and Melinda Gates Foundation on the malnutrition project. And we work with supplement companies, CPG companies, ingredient companies. So it's basically across academia and private companies. And so as an example, I just want to illustrate non-alcoholic fatty liver disease which we addressed as a first one in 2017. A collaborative study started. And the question is really, can plant-derived bioactives tackle one of the world's most pressing chronic health conditions, right? So NAFLD is highly prevalent. 1 in 4 individuals across the globe suffers from some degree of NAFLD. It can go to NASH, non-alcoholic steatohepatitis, which can be very fulminant hepatitis with liver failure. And so this is increasing rapidly across the globe. The HNF4 alpha, which is hepatic nuclear factor 4, it's a transcription factor of the nuclear receptor family, and it is a master regulator of lipid and glucose metabolism in the liver, and it's associated with several metabolic diseases. So a few years ago, one of the founders really had an interest in that, and they really started focusing on that target. So the target is really use the Forager artificial intelligence tool to identify novel HNF4 alpha agonists in dietary plant sources. And the Forager tool is basically described here, but it's a computational model, and with the ability to scan the plant kingdom for the best sources of a given high-value bioactive. And it employs a methodology to make increasingly accurate predictions of a plant's ability to produce specific metabolites and knowledge of evolutionary relationships amongst plants. And then based on that evaluation, compounds are chosen for the next step of in vitro validation and eventually also clinical studies and safety and toxicological studies. So we answered 3 questions. Firstly, what are the metabolites in the plant? So coming back to metabolomics, right? That's really a key question. So there are a large number of plants described. What's in the plant? And then what do each of them do for health? So we have also access to large databases of all kind of health targets, receptors, enzymes. Also, the scientific literature, you can develop your hypothesis as a target, and then what is the best compound source? So what is the plant source that would be best viable? And it can be on all kind of parameters: economic, sustainability, cost, and all those factors. So then how do we query Forager? So a computational model was developed to screen phytochemicals that would interact with HNF4 alpha, specific computational model. And we queried the database of the plant compounds in that database to identify potential agonists of HNF4 alpha. And out of this, 2 hits, or basically 1 hit, came forward, which is N-trans-caphoryltyramine, NCT, and N-trans-furolyltyramine. So they're very alike molecules and structurally very closely related. Also, the tool was able to produce a viable compound lead targeting this agonist in 3 months versus 3 years, which is the more regular time needed. And even, in this comparison, in 3 years, in the normal high throughput screening methods, nothing was found, right? So we were able to have a hit rate here of 3.7%. And actually the hit rate has increased over time. So it's a machine learning tool, right? Over time, it becomes more and more specific. The database grows of plant compounds, so you have more to choose from, and other factors would go into there. So in 3 months, a viable lead was identified, which was the molecule I just described, and you see them here on the right. So they're very closely related, these 2 molecules, with a methyl group just on the carbon on the top left. So the next question is to identify plant sources and perform preclinical testing as a first source. So the tool identified NCT in an upcycled source, which is called hemp hulls, with high speed and efficiency. So we identified basically 2 sources, which is black peppercorns. First, a lot of plant sources were identified, and these all contain these compounds. But then we focused on a fewer, which had a very high level of these compounds. And from there, 2 food sources were identified, which were, it's part of the food chain, because in the end, we'd like to have a safe source that can be readily consumed rather than a source that is very difficult to get, right? And that we don't have, data on consumption patterns for them, right? So this is how we got there. So the hemp hulls is actually, it's a waste product, or it's an animal feed product, but it's low cost. And so there's a lot of products out there which are basically what we call food waste, but you often, having worked in the food industry for quite some time, you see that often the most valuable things don't find themselves into the foods, and they end up somewhere else. And I think, this is a real opportunity. So this shows you some preclinical studies that were done with the group of Sanford Burnham Presbyterian Research Institute in California, and with Dr. Fred Levine's group. Several studies were done, but this is just, I think, a nice demonstration. And so on the left, you see a study where mice were fed normal chow, high-fat diet, or high-fat diet plus NCT for 10 weeks. And then they were looked at for the liver, and they were checked for their body weight over time. So on the left, you see that in the normal chow, you have like a normal size liver with a lot of redness. High-fat diet, the liver becomes almost twice the size. But when the mice get high-fat diet plus NCT, their liver remains basically the same size, almost the same size, which you can see in the graph there on the graph in figure H. If you now go to the right side of this, you see here an expression of a liver section, and in the fluorescent green staining, you see in the normal chow, you see HNF4 alpha expressed. High-fat diet, HNF4 alpha goes down. With NCT, actually it goes back up again. And when you look at the fluorescence, you see it actually in figure B, you see it significantly going up. Very interesting, and to our surprise, you see body weight gain being reduced when NCT is added to the high-fat diet. So you give these mice a lot of calories, high-fat diet. In the normal chow, you see a normal weight increase, but with NCT, actually, there's 25% less weight gain. Also, the food intake was the same between the groups, and there was also no more fat secretion in the stool. So that was an interesting check. And then you ask, where does it go? Well, you see actually an enhanced mitochondrial mass, which is described in another paper. So there seems to be something on metabolism happening, which needs to be figured out more. But so the conclusion is that Forager, an AI platform, identify two bioactives at superior speed and efficiency. They may be effective in nutritional and therapeutic management of NAFLD, which is highly prevalent. Furthermore, Forager identified the presence of these bioactives in an upcycled plant source, allowing further upscaling and clinical testing. We have a clinical trial ongoing now and plans for more. So clearly, this is an example, but it can be applied across, any benefit and any other target, basically. So there's a real opportunity for artificial intelligence such as this, which includes metabolomics, which includes machine learning, which includes all kind of input in an algorithm that improves itself over time. It accelerates the discovery and introduction of bioactive compounds, addressing unmet needs for both consumer and patients. So, that 99% that's out there, there's an enormous opportunity. So that's what I wanted to share. Thank you.
So, any quick questions? I think. Thank you. Oh, please.
So once you discover something that works, then what? How do you get it into?
Sorry?
How do you go from the discovery to a product that would be available for humans?
Yeah, so we have firstly, clinical studies are extremely important. So for this, we have a clinical study ongoing. We're doing safety, extensive safety testing, toxicological testing. So
But your company's not going to be the manufacturer of the drug, is it? Or
well, we're not targeting drugs specifically.
okay,
it's
or
real.
So you would actually be the producers then?
Yeah, it's real a nutritional focus, although we can also go into pharmaceuticals with this. But yeah, in the end, we do upscale. We do develop pilot scale to actually market the bioactive in the
Because
market.
some of your partners are producers of commercially available.
Yeah, it's business to business. Yeah, yeah, yeah.
Great talk, wonderful. How do you disentangle the potential for compound-compound interaction within the natural product? Because are you testing isolated products, or you are clustering products with your artificial intelligence?
Yeah.
Because that goes to the matrix of the food, right?
Mm. Yeah. So we test we have the opportunity to test the synthetic compound and the extract itself, right? So in the end, it's to do about the extract in the human being, right? Because that is what we're testing in the clinical study.